Good morning, everyone. We'll go ahead and get started this morning. It's my pleasure to introduce a friend and colleague, Mitch Phelps, from the Ohio State University College of Pharmacy. Mitch and I have known each other for a while now, back in the clinical pharmacology realm through various organizations and collaborations through the years. Mitch is an Ohio native and certainly is someone who I think he'll show you with his presentation today, an incredibly good thinker. He's also a great collaborator and just an overall good guy. He got his PhD in biophysics from the Ohio State University and has been there for many years now, and is certainly well regarded by many of you who know him, from those of you who have migrated from Columbus down to Atlanta, but also many others. He's certainly well funded, has just about 100 peer-reviewed publications over his time period at Ohio State. And one of his R01s is looking at the idea of transporters and their role clinically in a variety of different case examples. And this morning he's going to talk to us about the work that they're doing at Ohio State on IMiDs and the role of their transporters in IMiD resistance. So Mitch, thanks and welcome.
All right. Thank you very much for the introduction, Dr. Harvey. Thanks everyone for giving me an opportunity to come and share some of the work that we've been doing at Ohio State. As the title of my talk implies, and as Donald just mentioned, we've been doing quite a bit of work with IMiDs throughout their development at Ohio State, and this story talks about the role, or at least the potential role, of drug transport and overall disposition as well as resistance for IMiDs. And so I'm guessing many of you know, or maybe all of you know, what IMiDs are, but just in case, this is sort of an introductory slide. So the molecule on the left is thalidomide, and thalidomide, of course, is the textbook example of how not to do clinical drug development, unfortunately. And the other two compounds, lenalidomide and pomalidomide, structurally very similar, and I've highlighted in red here the differences. So, and I'll talk a little bit about how these, what appear to be subtle differences, can have a fairly big impact on the differences in the pharmacology of these agents. And I'll also mention that throughout this talk, you might hear me refer to either lenalidomide as Lena or pomalidomide as Poma, just to save a few syllables throughout the talk here. This is a time frame of development. I put this up here just to kind of point out that the clinical use of thalidomide it started back in the 1950s and the 1960s in Europe, and that's actually when it was discovered. It was a teratogen. Unfortunately, it was given to pregnant women as an anti-emetic, and it was discovered in the 60s that thalidomide, early 60s, that thalidomide was the cause of the teratogenic effects. Actually, though, because of some interesting properties thalidomide had, it continued to be evaluated and developed for a variety of other indications. And so a couple other things I'll point out here is that actually in 1998 in the US, thalidomide was approved for use in EL or ENL or leprosy. And right after that, a phase 2 trial of thalidomide in relapsed and refractory multiple myeloma was also started. So really in the late 90s and in the early 2000s, there was a lot of development both with thalidomide and also one of the other agents, lenalidomide. A lot of it was in multiple myeloma and also in myelodysplastic syndromes, in which the drug lenalidomide was approved in 2005. And so I'll talk a little bit about some of this over here in a second as well, but I guess this is at least 50 years, maybe a little bit more than 50 years, where we were using these agents in the clinic, but really didn't understand fully the mechanisms until just recently. And so if you look at the general pharmacology of these agents, again, some of the interesting properties, both from a cancer aspect and also from non-cancer aspects, they're anti-inflammatory, anti-angiogenic, anti-proliferative. They co-stimulate T cells, they activate NK cells, they disrupt some interactions in the microenvironment, inhibit TNF-alpha production. The toxicity profile, not too drastic outside of the teratogenic effects, of course, that's managed, but the myelosuppression, neutropenias, some thrombotic events. Tumor flare seemed to be kind of unique in CLL, so when we were looking at lenalidomide and CLL, this sort of came up as somewhat of a unique toxicity. There is an increased risk for secondary malignancies. Also, some neurotoxicity, and I'll talk a little bit about that, and that gets back into how these different agents are fundamentally different in terms of their pharmacology, and also skin rashes. And I'll point this out. So I'll talk a little bit about myeloma because that's where these drugs have been most prevalently used. But just wanted to point out and have this slide that shows current therapies in myeloma. And essentially there are two paths. There are patients who are transplant eligible, patients who are transplant ineligible. Both get induction therapies, and of course, in transplant there's maintenance therapy as well. Just showing here that these agents, all three of these, show up really throughout the treatment regimen for myeloma. So very important in terms of management of this disease. And I'll also highlight here another drug that's very prevalent, and that's dexamethasone. And I'll talk a little bit more about why that's potentially relevant here shortly. Okay, so as of yesterday, I update this slide every time I talk about the work we've been doing with these IMiDs. As of yesterday, more than 770 clinical trials have been initiated with lenalidomide in particular, and this sort of ranges a lot of different diseases, multiple myeloma certainly being one of those, but various cancers, both hematologic and solid tumor malignancies, single agent and combinations. And more recently, sort of the standard of care, which is lenalidomide and dexamethasone, is being put in combination with some of the newer antibody therapies in multiple myeloma and other diseases as well. Also, these are early stage maintenance or even refractory or retreatment settings. And also there's been some work in children with these as well. So just to kind of give you a sense of the kind of broader interest with respect to IMiDs across a broad array of diseases. Okay, so I mentioned it was just recently that we discovered what the mechanism, or the direct target anyway, and later the mechanism of action for IMiDs was. And so this is a paper that was published in 2010 in Science, and identification of a primary target of thalidomide teratogenicity. And so this paper talks about a protein called cereblon, which is just a year later, basically a group of investigators had looked at cereblon expression with respect to the anti-myeloma effects of IMiDs, and sort of confirmed that does appear to be the right target. And then within a couple years after that, some additional work was done to show that the cereblon-dependent destruction of a couple of proteins, Ikaros and Aiolos, are really critical for the activity of IMiDs in myeloma. And the same year, two groups came out with very similar findings that sort of solidified this mechanism. And this paper also kind of highlighted the T-cell co-stimulation and with respect to the Ikaros and Aiolos proteins as well. Okay, so not sure if everybody can see that, and I apologize if it's a little bit small back there, but so on the left side, I'll just kind of walk through this mechanism. On the left side, we've got this, so cereblon is part of an E3 ubiquitin ligase. And so in the absence of an IMiD, you have this E3 ubiquitin ligase, which can ligate its targets. But it also has this auto-ubiquitination component as well. So cereblon ubiquitinates itself, and that leads to its own degradation. And without IMiDs, you have these two proteins again, Ikaros and Aiolos, which are expressed essentially they're involved in the expression of IRF4, which is an important protein in myeloma. IRF4 then is transcription factor that's sort of involved in the expression of MYC. And so without an IMiD, this process is happening, and these are tumor growth pathways that are activated. But over here in the presence of an IMiD, what appears to be happening is that ubiquitination process is blocked, and therefore that seems to stabilize this complex. And it also seems to increase the affinity of this complex for these Ikaros and Aiolos proteins, which leads to their degradation, which then stops ER4 transcription as well as MYC production. And it also releases IL-2 production as well. So this was really just kind of worked out in the last maybe 5 to 7 years, how these were working despite our use clinically for the last maybe 20 years in multiple myeloma. Okay, and I'll highlight here too that all three of the structures that I showed you, so thalidomide, lenalidomide, and pomalidomide, they all have similar affinities for cereblon, yet the potencies are different. And so pomalidomide is the most potent, followed by lenalidomide. There's maybe a 10-fold factor difference there. And then both of these are pretty significantly more potent than thalidomide. And so some of the things I'll talk about with respect to transport, we think that transport could be playing a role in some of this different potency as well. Okay, so we set out, so obviously development of these agents was already well underway before I got involved, but I got involved probably around 2007, 2008, so maybe about 10 years ago at Ohio State, and we were looking at the use of primarily lenalidomide in multiple diseases, multiple myeloma being one of them, but other diseases as well. And so here, pharmacology, just PK from the literature. When I started looking at this, frankly, this looked like a very boring drug from a pharmacology standpoint. It has very high oral bioavailability, very rapid absorption, linear pharmacokinetics, dose proportional over the ranges that it was being used clinically. Low plasma protein binding, wasn't a substrate inhibitor or inducer of CYP enzyme, so it's not even metabolized. It's pretty much almost entirely eliminated as the unchanged drug in urine. Renal impairment certainly had an impact on the pharmacology or pharmacokinetics, at least of lenalidomide. And there were what was known at the time similar PK parameters between adults and children. And then when we looked in other sources, so at that time, just sort of digging into some of the EMA documentation as well as FDA documentation that was being produced at that time, some other information, so relative oral bioavailabilities in different species, distribution. This was kind of interesting, extremely low brain level, so I'll talk a little bit more about that shortly. Again, metabolism, very, very limited metabolism. I think I just said no metabolism, but in fact there is a very small amount of metabolism that happens primarily on this amine group. And then all three of these compounds, all three of these IMiDs, so can undergo hydrolysis. So there's actually a switching back and forth between an R and an S enantiomer. And so that's been very well studied, very well characterized, primarily with thalidomide. We did quite a bit of work trying to understand, is that important with respect to lenalidomide as well. Okay, so there were still some questions that we thought we could try to help answer at that point. And so for example, in humans, what happens to that other third of the drug that's not found in urine? Are there metabolic pathways that hadn't been identified yet? Is there a lack of absorption, biliary excretion? What were the mechanisms for renal excretion of lenalidomide? So we were already looking at transporters that may be involved. We knew that glomerular filtration was actually a little bit higher than the clearance of than renal, or I'm sorry, glomerular filtration was actually a little bit lower than the renal clearance of the drug, and therefore we thought there could be something interesting here to look for. And then also just sort of understanding tissue distribution, and as this is being evaluated in multiple diseases, trying to understand how this drug is reaching the tumor compartment in these different diseases. And so this was one of the first data sets that we had generated. So a clinical data set where is a very small study. I think there were about 21 patients where we had pharmacokinetic data with lenalidomide, and this was in multiple myeloma patients. In this case, we were looking at a combination of lenalidomide with temsirolimus or CCI 779. And what we saw, again, for me, kind of when I was first starting out in clinical pharmacology, and this is what all of us want to see is something interesting, and this looked pretty interesting to us. Essentially what this says is that as you increase the dose of temsirolimus, what you see is an apparent decrease in clearance for lenalidomide. Now, again, the caveat here was we only had 2 dose levels, very small number of patients in this group, of course. But we saw a trend here that looked kind of interesting. With respect to, if we looked at the potential drug-drug interaction the opposite way, we saw really the same thing. As we increased the lenalidomide dose, there was maybe what appeared to be a trend for decreasing clearance of temsirolimus. And so this was something interesting that we kind of latched onto. And so when we started asking what could possibly be happening here, how could there be a drug-drug interaction? Again, lenalidomide is a pretty boring drug, I would say. Overall, it's not a substrate inhibitor, inducer of CYP enzymes. Some interaction observed between lenalidomide and digoxin in previous clinical data. And that digoxin is a model P-glycoprotein substrate, so that gave us a little bit of a clue. And then also, with respect to temsirolimus, it's a known PGP substrate and inhibitor. So what we wanted to look at here was a potential role of P-glycoprotein in this apparent drug-drug interaction. So we went back to the lab and we started doing a variety of different studies to investigate this. And essentially what we observed was that lenalidomide is in fact a PGP substrate. And so these are model cell lines, HL60 versus the vincristine-resistant version of HL60 BCR. And so if you just look at the difference in uptake of lenalidomide in those 2 cell lines, you see a difference visually and statistically, the difference is there. And this so suggested, of course, that PGP was actually playing a role in absorption and efflux out of the BCR cells. You could reverse this, so verapamil is a very common agent that we use in vitro to look at substrates of P-glycoprotein. And we saw a little bit of an effect with verapamil, but actually in vitro, when we took temsirolimus and added that to culture, we could essentially restore the uptake level back to the level of HL60, which does not express P-glycoprotein. And so this was a pretty big clue for us that there was a clinical interaction happening here. This is just looking at sort of another cell system. So these are uptake studies, and so this tells you how much drug is actually getting into the cells versus these are actually transport assays. So looking at transport across an epithelial membrane layer. And here we're using MDCK2 cells, a very common model for conducting transport studies, and then MDCK2 cells that overexpress PGP. And so this is an apparent permeability calculation, and of course, we're doing something called apical to basolateral or basolateral to apical efflux. So we're looking at transport in both directions across the membrane. And when we see this spike in the PGP expressing cells, that tells us again that PGP is playing a role here in the transport and ultimately the efflux of lenalidomide. So then we started asking the question, how important is this with respect to drug resistance for the immunomodulatory drugs? and also what other transporters may be involved in immunomodulatory drug uptake and efflux. Yeah, so Celgene owns all these immunomodulatory drugs. They were not happy. I can't say I blame them. So they had just gotten approval relatively recently at this time, and here we were presenting some data that suggested there could be clinically relevant drug-drug interactions. And so it was kind of interesting. We published this paper, and very shortly after that, they published a kind of a response to the paper that said the evidence doesn't support clinically significant drug-drug interactions via P-glycoprotein. And there's a good lesson in this that I'll come back to here toward the end. But needless to say, they were not happy with what we had found and what we had published here. Yeah, so what might be important, both from a pharmacokinetic and drug-drug interaction perspective? Okay, so what other substrates, inhibitors, or inducers of P-glycoprotein might be relevant clinically? So we started out with cell line studies with other drugs, and so I had mentioned earlier that dexamethasone, of course, it's an important agent in treatment of several different diseases, but certainly in myeloma. And we are combining dexamethasone with immunomodulatory drugs when we give it. And dexamethasone is a known inducer of P-glycoprotein, so it was kind of an interesting question to pursue, we thought. And then we also wanted to do some in vivo studies looking at possible drug-drug interactions in a PGP knockout mouse model, MDR1 AB minus. And then also try to understand this hydrolysis and GI tract pH changes. So I had mentioned these drugs are chemically unstable, they hydrolyze, and how might that be impacted if you're giving proton pump inhibitors, for example, since these are oral agents. And then we had several other ongoing clinical studies that we were conducting, and some of those were in combination with other drugs. So we had some opportunity to look at possible drug-drug interactions in the clinic just with respect to the studies that were already ongoing. So I think I have one slide later where I'll mention a study where we were combining with idarubicin, which is also a PGP substrate, so we had an opportunity to take a look there. Yeah, so with respect to the question of dexamethasone and inducing P-glycoprotein, we started out looking in several cell lines, and we were kind of thinking of this really from the whole body perspective as opposed to drug-drug interactions or drug resistance in the tumor at this stage, just based on what we had seen. So we wanted to ask the question if there is going to be a drug-drug interaction, where is it occurring? Well, lenalidomide is known to be renally excreted primarily, and so looking in renal cells and also in liver cells made a lot of sense. And of course, we wanted to look in myeloma cells as well. So we kind of set out on this small project to kind of understand what might be happening with co-administration of dexamethasone and lenalidomide in this case. And so these are different kidney cell lines. And so what you can see, so this is DMSO control, lenalidomide alone, dexamethasone alone, or the combination of lenalidomide and dexamethasone. So that's repeated throughout these next few slides here. But what we saw was that there was some variability between the different cell lines. In some cell lines, we could pretty drastically induce P-glycoprotein expression, versus in other cell lines, we really didn't see any impact whatsoever. And notice too that expression was about the same whether we were giving dexamethasone alone or in combination. And then a couple of different liver cell lines, just somewhat of a contrast. So liver, both liver and kidney express P-glycoprotein, maybe more so in the liver. And some of the cell models we looked at for liver, they were already expressing high levels of P-glycoprotein. And so when we looked at mRNA, we did see small increases in mRNA expression once we gave dexamethasone or the combination. but overall there was already a lot of P-glycoprotein there, so there wasn't a whole lot of increase that we observed once we gave the combination above the baseline levels. We also wanted to understand the time course for induction of ABCB1, which is the gene for P-glycoprotein. And so this is another kidney cell line that we were evaluating. Again, the same order, DMSO, lenalidomide, dexamethasone combination, going from the left to the right. And what we saw was over time, you got further and further increases, and we kind of said maybe 72 hours is about where the max is happening. And it was again pretty drastic and pretty significant increase in P-glycoprotein. One of the reasons we wanted to do this time course study was in our animal studies, we're trying to understand when to dose and exactly how to dose these animals, and when it would make sense to go back and take a look at PGP levels in the animals. We also confirmed that with that dexamethasone induction of P-glycoprotein, that we saw a functional effect, meaning that with no induction, we saw a baseline level of lenalidomide uptake in these cells, and then once we induced with dexamethasone treatment, we then saw the decreased uptake as we would expect with a large amount of P-glycoprotein expressed. And then we came back with temsirolimus and tried to inhibit that, maybe a little bit of a inhibition of PGP in the system, but not much, and a lot of variability there as well. And then verapamil didn't seem to do a whole lot. But nonetheless, we saw what clearly was a functional effect of combining dexamethasone with in this case, lenalidomide in this cell system. We also went back and looked at a variety of myeloma cells and did similar work. This slide was just to show that so these are relative mRNA levels, just to show that across a variety of different cell lines with multiple myeloma, we were seeing significant differences in PGP expression. Again, this isn't groundbreaking, of course. We would expect this when you look at different cell lines that you may see something like this, but it gave us at least some idea that PGP could be important with respect to immunomodulatory drug resistance in myeloma. So I'm going to jump now to in vivo studies. So that was just kind of a bit of some of the in vitro work we were doing. And at the same time, we were, again, trying to understand overall disposition of the different IMiDs, mainly focused on lenalidomide. And this was a study that we did, published about 5 years ago, I guess, in mice. So here we were really trying to understand if we were going to study these immunomodulatory drugs in mice. We didn't know at that time that you really couldn't do that because we later figured out, as others did too, that mouse cereblon doesn't respond the same way that human cereblon does. But when we were trying to study different diseases in different mouse models, we wanted to really thoroughly characterize overall disposition of these drugs within mouse models. And so the main point of this slide is to show you that when we looked at lenalidomide across many different tissues, of course what you see is a range of exposures in those different tissues, which is not surprising. But one of the things that was interesting here was we couldn't even detect brain levels until we got up to some of these higher doses, and just noticed that with all the tissues, brain has the lowest normalized concentrations across all the tissues. So again, maybe not necessarily surprising, but kind of interesting. We then went into, so I mentioned the knockout mice. So these mice have the knockout of 2 genes. So in the mouse, they have 2 genes that that effectively work as P-glycoprotein. And so these are mice that have both genes knocked out. And what we expected to see in these mice was pretty significant differences in pharmacokinetics. Concentration time profiles here. So this is wild type intravenous dose, and then a PGP knockout with intravenous dose, and then down here we have an oral dose set of profiles as well. And so with the IV dose, we really didn't see massive differences, pretty modest differences. There were some effects. So AUC increased a little bit, therefore clearance decreased a little bit. And then when we gave an oral dose, we saw maybe slightly bigger increases in AUC in the PGP knockout mice, but it wasn't quite as drastic as what we had maybe anticipated that we might see. At this point, we also started working with one of the other IMiDs, Pomalidomide, and again did the same study in the knockout mice, again wild type versus PGP knockout IV and oral. And here we saw what relative to lenalidomide was a bigger effect with the PGP knockout. And this was kind of interesting because we saw it much more so in the IV dose versus the oral dose. And again, both of these are orally administered agents in patients. P-glycoprotein is expressed in the intestinal or in the gut, and therefore we thought we might see some bigger effects overall here. But nonetheless, we did see some effects, which again kind of led us to believe that maybe pomalidomide also is a PGP substrate. So we're starting to do some in vitro studies at this point as well. Actually, shortly after we completed these studies, a group had published this paper. So pomalidomide, they were evaluating cytochrome P450 and also transporter-mediated drug-drug interaction potential in vitro and also in some healthy subjects. And so this actually in part was a response to some of the work that we had published before where we had seen what we anticipated was a clinical drug-drug interaction with lenalidomide. And so just to highlight some of the things that are stated in here, they looked at a panel of different transporters, P-glycoprotein being one of those, BCRP, which is another multi-drug resistance transporter, and then some SLC transporters that would be relevant either from an oral absorption standpoint or from a liver standpoint. And they didn't really see any inhibition of any of these transporters. And then pomalidomide, I don't think I mentioned this before. So lenalidomide is different from the other two compounds because the metabolism is very, very low. But pomalidomide, there's a significant amount of metabolism that takes place with pomalidomide and also with thalidomide. And they did indicate in this paper it's a PGP substrate. They also said that pomalidomide appears to have low potential for clinically relevant drug-drug interaction and unlikely to affect the clinical exposure of other drugs. So nice bit of work there by this group. Going back into our mouse data and the knockout mice. So what I showed you a couple slides ago was the plasma exposure data. And we wanted to look in the brain tissue as well of these mice. And again, thinking that the brain is going to be a place where if PGP is important in the disposition of lenalidomide, this is probably the tissue where we would see it. And so on the top here, I've got data for lenalidomide. On the bottom, data for pomalidomide. And the way this is shown here is plasma versus brain of the knockouts in the dark, and then plasma versus brain of the wild type. And so one thing that you notice here is that if you just compare the plasma levels between the wild type and the knockout, there's not a whole lot of difference. Again, we saw a pretty modest increase in lenalidomide exposure in the brain with the PGP knockout mice. but it wasn't nearly as much as what we would have anticipated based on the work we had done up to that point. The other thing that we noticed is that the brain penetration for lenalidomide was pretty much equivalent in both the wild type and the knockout animals. And so what that told us was that, and not only was it pretty much equivalent, it was still very, very low. And what that told us was that there is something preventing lenalidomide, obviously, from getting in the brain, but it's not P-glycoprotein, it's something else. And interestingly, pomalidomide, very different with respect to brain exposures. So here we had brain exposures that were maybe about 10% relative to plasma. With pomalidomide, they were just about equivalent to plasma. So this told us that pomalidomide actually can get into the brain pretty readily. So there's a difference between these two drugs in terms of their brain penetration, at least in the mouse, and this permeability difference appears to be independent of P-glycoprotein. So looking in the literature, there was a paper published in 2012 where a group was looking at both plasma and, so this was cerebrospinal fluid, CSF, pharmacokinetics of both thalidomide and lenalidomide in non-human primates. And a lot of data in the tables. I'll just kind of highlight the important things over here. So these are AUCs, AUC ratios of CSF versus plasma. And so when you look at thalidomide, you get numbers between 36 and 50 versus lenalidomide, essentially between not detectable and everything else looks like it's 11. So again, thalidomide may be more similar to pomalidomide, does appear to get into the brains, at least in non-human primates. So again, pointing to a fundamental difference in lenalidomide overall disposition. Okay, so this was, there's the date. So 2011, this paper had come out. So I had mentioned before that the mainly lenalidomide at that time was being evaluated in a variety of different diseases. Some of those even being CNS tumors. And so this was a trial of lenalidomide in pediatric patients. They did see some partial responses in this trial, but based on the data and the expectation that there's actually low penetration in the brain, does that mean that it doesn't make sense to try to develop an agent that can't very well penetrate the brain for brain malignancies? In some cases, maybe, but so this was just kind of an interesting observation. So then why, if both lenalidomide and pomalidomide are substrates of PGP, do they have such drastically different brain permeabilities? So the blood-brain barrier, of course, is made up of a variety of different proteins, and PGP is not the only efflux transporter. BCRP is another very important transporter in the blood-brain barrier. And so we wanted to ask the question, is BCRP also involved in lenalidomide or immunomodulatory drug disposition? And so again, these are MDCK cells. This is intracellular drug levels. So this is uptake versus the efflux study. And we saw pretty similar effects with BCRP. So this is MDCK wild type versus PGP overexpressing versus BCRP overexpressing. And again, we see the decrease when we go from the decrease in uptake when we go from the wild type to either PGP or BCRP overexpressing. And with the transport studies, data is a little bit messier here, but we saw similar trends as well in terms of the efflux ratios being different when you had cell lines that were overexpressing BCRP. And so the conclusion we came up with here was lenalidomide is in fact a BCRP substrate as well. We also had data on pomalidomide. It's a little less clear than this, a little messier, but we came to the same conclusion that pomalidomide too is probably a substrate for BCRP. Yeah, so this, just to show you as well, so as we're doing the uptake and transport studies, we're calculating something called an apparent permeability. And another interesting thing that we saw here was when we looked at lenalidomide relative to pomalidomide, you can just think of apparent permeability essentially as how easily does this agent get across the membrane or the epithelial layer. And so lower values means less drug is crossing. And we were seeing these very, very drastically different values between lenalidomide and pomalidomide. And so these numbers, for those of you that can't see, basically 1 and 2 versus 246 and 147. Okay, so again, in this in vitro assay, very, very drastically different capabilities for lenalidomide relative to pomalidomide to cross the membranes. So there's another assay. It's called the parallel artificial membrane permeability assay. And so in this assay, you essentially remove the cell system. So you're getting rid of all transport proteins. And you're just asking the basic question, how easily do these agents cross an artificial membrane? And you can use different lipids to create these membranes. And basically what we were seeing here was that there really was not a big difference if you look at lenalidomide values versus pomalidomide values. We were coming up with maybe twofold higher values for pomalidomide, but that nowhere near explains the drastic differences we are seeing in the cell system. So again, kind of raise the question of what's happening here and why are we seeing these differences. I will show you this data. This is actually from a clinical trial. So this was a study that we were doing. I had mentioned the study with idarubicin. So this data actually came from that study. The PI was actually Bill Bloom. And we were looking at patients who were either treatment naive in blue, so the ones on the left, versus patients who were who had relapsed refractory AML. And those are highlighted in red on the right. And here we were looking at the expression of 3 genes, ABCB1, ABCG2, and then also cereblon during the treatment. And so these patients were getting lenalidomide for the first 5 days of treatment alone. So we were looking at expression day 0 versus day 5, so just after 5 days of therapy, essentially. Again, treatment naive versus previously treated. And so one thing that stands out, again, maybe not too surprising, that each of these genes is more highly expressed in the previously treated patients. So these patients are already relapsed or refractory. So these multi-drug resistance genes are already more highly expressed. And then another thing that we noticed too is that there didn't seem to be a whole lot of difference between day 0 and day 5 with at least these 2 transport proteins. When we looked at cereblon, however, it was a little bit different. Again, maybe a little more expression in relapsed and refractory patients. But we were seeing a difference actually between day 0 and day 5. So again, these patients were just getting lenalidomide, and it looked like cereblon was kind of responding to that treatment and maybe being increased in terms of expression. So again, a little bit of clinical data that suggests that maybe actually even in the tumor cells, so I forgot to mention, I think, that we were collecting both bone marrow and peripheral blood mononuclear cells, and that's where this data was coming from. So won't bother to read all this, just kind of summarizes what I had mentioned, but essentially we were seeing, I guess first of all, that there was evidence that the fact that lenalidomide and pomalidomide are PGP substrates maybe is relevant clinically, and certainly seeing some kind of interesting things when we were combining other drugs, including dexamethasone, where you could increase PGP expression. we were seeing these differences between lenalidomide and pomalidomide in terms of their overall disposition and especially brain penetration. And then in the in vitro assays, kind of these differences in the ability of these compounds to pass membrane. So what we ultimately conclude was other transporters must be involved in lenalidomide and or pomalidomide disposition. So in addition to PGP and BCRP, there are other transporters out there that we have not identified yet and others have not yet identified as well. So a lot of what I showed you, work that we've done a while ago, we haven't published it, but Celgene ended up doing kind of a series of studies, and I guess I'll say that they did a very thorough job, I think, in kind of asking this question and looking at how relevant is this, probably on their own behalf, although maybe the FDA had some input there in terms of dictating what needed to be done. But so the lesson that I had mentioned before was that really with all the work that we did, both in vitro and then in the animal models, we kind of came to the same conclusion that Celgene came to, and that was that first bit of data that we got in the clinical trial where it looked like there was this drug-drug interaction, and that data led us to discover that lenalidomide is in fact a PGP substrate. With the data that I showed you in the mice, how PGP didn't really seem to impact the disposition, all the data that they had generated, the clinical trials where we were looking at drug-drug interactions, we just really haven't seen anything. And so I think that their due diligence here was really kind of spot on. And so the lesson that I learned from that was what we all learn in classes, and then as we get out there and start to work with real data, that those small trials where you have only 20 patients, even when you see something that looks extremely obvious, may not be so obvious. In your interpretation of that data, you really just have to be very careful the way you interpret it. So I'll pass that along, and I'll admit that we probably jumped the gun a little bit there on maybe some of the things that we said in that initial paper. But and then so these last few slides here, I just want to show you kind of a shift in focus. So this was a paper published in 2013. Protein called CD44 was found to be important in cell adhesion and maybe mediating resistance to lenalidomide in multiple myeloma. And we published a couple of papers here where we identified CD44 also as a serum marker in multiple myeloma patients who were relapsed and refractory to lenalidomide. And this AR42 compound, so it's an HDAC inhibitor, homegrown at OSU. So we were involved in the preclinical development of this, and then in the first in-human studies, and we've got a couple of other trials still ongoing with this. We discovered that this actually decreases the expression of CD44 and can sensitize myeloma cells to lenalidomide. So a group of people, Craig Hoffmeister, Flavia Piccioli, were leading that work. So this CD44 hyaluronan acid receptor binds to hyaluronan and other extracellular matrix proteins, and it's a marker for stem cells commonly overexpressed in drug-resistant tumors. So it wasn't just multiple myeloma. You actually see this protein show up in a lot of refractory diseases. And also it interacts with various membrane proteins, including transporters, receptor tyrosine kinases, and others. So there was a series of papers that were published over the last several years showing that CD44 can interact with and even stabilize both P-glycoprotein and BCRP. So that kind of got us thinking maybe what we need to be focusing on. I'm sorry, this is a very busy slide. Maybe what we need to be focusing on, instead of kind of looking at whole body disposition, going back to the more classical way that we think about transporter-mediated resistance and cancer, and that's looking at the tumor cell. And so this slide, again, very busy, and I won't try to explain everything here, just sort of represents our current model and the way that we're approaching this right now. So we're conducting clinical trial in conjunction with Craig Hoffmeister and Flavia Picciori, and we're looking directly at this question. So looking in the tumor cells themselves and the plasma cells, are the cells that are overexpressing CD44, do they in fact have higher surface expression and higher functional expression of these transport proteins, PGP, BCRP, and others? And is that impacting the ability of lenalidomide to get into these cells? So we're treating patients, we're collecting samples now, and we're starting to do some of these studies. And this, I guess I'll just say, with all the data that we've accumulated over the last decade with lenalidomide, we've got quite a bit of combined data, and we're putting some papers together that are combining data from different studies, looking at the pharmacology of lenalidomide and soon pomalidomide as well. And I'll just mention AR42 as well. So one of the interesting things with this trial is that we're actually collecting bone marrow samples from patients after they're being given their first dose of lenalidomide, or I'm sorry, in this case it's pomalidomide and AR42. And we're collecting those at various time points over a 24-hour period so that we'll get a first look at bone marrow exposure of both of these agents. So this is next to last slide. So in search of the IMiD transporters, a paper just came out a couple years ago where the group had used RNAi screening to identify genes that were relevant for sensitizing multiple myeloma cells to lenalidomide. And they came up with a variety of genes, and what we noticed was that there was a handful, about 10 different genes that were transporters. And so we're now currently probing these, and we're also doing some of our own screening to look at other transporters that could be involved. So again, we know from all the data that I just presented that there are other transporters that have not yet been identified that are important, at least from a disposition standpoint. Whether or not those are going to play a role in resistance, that's still yet to be determined, but this is where we are right now. And with that, I would just like to acknowledge a lot of the folks who created the data, so lab members, and then we have a huge number of collaborators, and many of those are clinical collaborators, including John Burr, especially Bill Bloom, I mentioned, Craig Hoffmeister, and then various funding sources. Some of this came from Leukemia and Lymphoma Society, Eli Lilly, and then also some funding from the NCI. And so with that, I'm happy to take any questions.